You know, in the world of plants, there's a reproductive strategy that is so strange, so elegant. It almost sounds like science fiction. It's the secret hiding inside pretty much every flower you've ever seen. Today, we're going to unpack the incredible puzzle of double fertilization.
All right, here's our roadmap. We'll start with the central puzzle itself. Then we'll look at the two separate missions of the male gametes. After that, we'll check out the ingenious power lunch this whole process creates. From there, we'll see how life's very first blueprints get drawn up. And finally, connect all this microscopic magic to the seeds and fruits we see every day. Let's get into it.
So, we're kicking things off with a real biological riddle. It's something that happens deep inside a flower's ovule, and on the surface, it seems to defy simple logic, but it's actually the secret sauce to the success of all flowering plants, or as they're known, angiosperms. Okay, so here's the puzzle. You've got one single pollen grain that kicks off two separate fusions, and those fusions involve a bunch of different nuclei. I mean, with all those moving parts, you'd think it might be called triple or even quadruple fertilization, right? So why the simple name double? What is the secret behind this really unique plant math? Well, the answer is all about understanding that the two male gametes are they're like two agents on two separate but equally critical missions. See, this isn't just one single event. It's two vital things happening at almost the exact same time. So mission number one is called Syngamy, and this is what we usually think of when we hear the word fertilization. A haploid male gamete, and haploid just means it has a single set of chromosomes, what we call N, fuses with the haploid egg cell. The result? A diploid zygote, or 2N, which now has the complete double set of chromosomes. This little zygote is the very beginning of the future plant. It is the creation of the offspring itself.
Okay, now for mission number two. This is where things get really, really interesting. The second male gamete, it completely ignores the egg. Instead, it makes a beeline for two other nuclei, which are called the polar nuclei, in an event we call triple fusion. That single N gamete merges with the two N polar nuclei, and poof, you get a single, triploid, or 3N nucleus, and that right there is the solution to our puzzle. Double fertilization is simply the name for these two totally distinct events, syngamy and triple fusion, happening together. It's like a two-for-one deal. One fusion creates the baby plant, and the other one makes its lunch. Let's talk about that lunch, because the result of that weird triple fusion isn't just some math curiosity, no way. It's an evolutionary masterstroke. It provides the brand new embryo with the perfect, dedicated food source, and we call this food source the endosperm. You know, this 3N number is the real game changer. Being triploid makes the endosperm a nutrient-packing powerhouse. Its one and only job is to store up starches, oils, and proteins just for the embryo. And think about how efficient this is. The plant only invests energy in making this food pack after it gets confirmation that the embryo has been successfully created. No wasted resources. It's an absolutely brilliant evolutionary strategy. Now, this endosperm doesn't just appear fully formed. It develops in a few different ways. The most common way, by far, is the nuclear type. Just imagine a tiny balloon where the nucleus inside just keeps dividing and dividing, filling the whole space with free-floating nuclei. It's only later that cell walls start to form around them. And while there are other patterns, the key thing to remember is that this free nuclear method is nature's most popular strategy for building this food supply. So while this little lunchbox is getting packed, the zygote from that first fusion is getting busy. It's dividing and growing in a process we call embryogenesis, and it follows one of two major blueprints for life. Let's take a look at how those blueprints are different. The two big blueprints are called Dicot and Monocot. Now, the easiest way to tell them apart is to look at their cotyledons, which are basically their first baby seed leaves. Dicots, think of a bean, have two of them. Monocots, like a kernel of corn, just have one. Oh, and monocots also give their baby shoot and root some extra protection, these tiny little sheaths that act like helmets, helping them punch through the soil safely when they start to grow. Now, here's a little detail that trips a lot of people up. That single cotyledon you see in a monocot, like in a grain of wheat or corn, it actually has a special name, the scutellum. It might look a little different, but really it's just a highly modified cotyledon. Its whole job is to help transfer all those nutrients from the endosperm over to the growing embryo. So we've seen the fertilization, we've seen the food supply being made,
And we've seen the embryo start to develop. Okay, now let's zoom out a bit and see how all these tiny, microscopic events lead to the structures we all know and recognize, the seed and the fruit. Right after fertilization, this incredible transformation begins. The entire ovule, which holds our embryo and its endosperm lunchbox, matures and becomes the seed. Its outer layers harden up to form the protective seed coat, and at the same time, the ovary wall that's surrounding it starts to swell and ripen, and it becomes the fruit. So, yes, the next time you eat an apple, just remember, you're technically eating a mature plant ovary. But what if you could get the fruit without any of the fertilization? Well, it turns out nature has a weird exception for that too. It's a process called Parthenocarpy. It's this fascinating thing where the ovary develops into a fruit all on its own, completely skipping the need for pollination, fertilization, and even seed development. And the most obvious result of this process is, of course, seedless fruit. The classic example that everyone knows is the banana. Yep, the bananas you buy at the grocery store are parthenocarpic. They develop without fertilization, and that's exactly why they don't have those big, hard seeds you'd find in their wild relatives. It's a natural quirk that we've turned into a massive horticultural advantage. And that kind of brings us to a final, much bigger question, doesn't it? We started with what seemed like a simple biological puzzle, but understanding it has literally allowed us to engineer the food we eat. So as our ability to read and understand these genetic blueprints gets better and better, you have to wonder, what's next? What other parts of nature's code are we going to rewrite in the future? Thanks for joining me on this one.